using RNA as a template for which is used to convert which is used to prepare the cDNA and used as a template in the real-time PCR for the gene expression. It includes the RNA as a template, then primer, DNA polymerase, dNTP, buffer and cofactors. So basically this four thing is more or less similar to what we are using in conventional PCR. However, the difference is that we are using RNA as a template and then we are using fluorescent molecules. Out of the several types of fluorescent molecule available for the real-time PCR, the most commonly used and most economic used is cybergreen, which I will be briefly touch upon. And the Tachman probe will be taken care of tomorrow by Dr. Venu. So cybergreen is an aromatic molecule with with a side chain, which goes and intercalates in the double-stranded DNA, minor group of the double-stranded DNA, and as a result of which the fluorescent intensity increases more than 1000-fold. As a result of which you can use the light source to excite this fluorescent molecule, which emits the light, and you capture in the CCD camera, which is reflected in the form of amplification curve over here. Now we will closely look at how this curve looks like. This curve looks something like in the regular curve, you will see that you have exponential as well as non-exponential form, which we will be discussing in the next slide. However, in order to check the real-time amplification, it is important to convert the y-axis into the logarithmic scale, and that enhance our visibility to notice the threshold value. So what this threshold value is, the cycle number where the fluorescent intensity is reaching to a threshold value. It means that it is subtracting all the baseline fluorescence, and it's visible and it's increasing in the linear kind of linear order in the exponential phase. So because of the ingredients which gets depleted as the cycle number increases, at certain point the PCR reaches to its plateau phase, that is also called as non-exponential phase. However, what we need to understand is that we have to calculate the CT value in the linear scale of the exponential phase. So CT value is the cycle number where fluorescence crosses the threshold line, and this threshold line we will see in the practical what it means. In this particular picture, what you can see is that every tenfold dilution, either the red one or the blue one, violet, and so on, every reaction is done on 10-fold dilution starting material, or you can say the sample. And then every 10-fold dilution causes 3.3 cycle apart. It means that the CT value difference between 10-fold dilution would be 3.3. And if that happens, it means that your PCR is having 100% efficiency or running in a 100% efficiency. It means that each cycle is increasing the gene with 2-fold, right? So what we can understand from this is that if initially you have the low copy number, then you can have the high CT value, and that can be called as low expression. If you have the high initial copy number, then low CT value, and that leads to high expression. Now, a quick review that you have the sample, you extracted the RNA, checked the quality of the RNA, converted the cDNA, and this cDNA using with the fluorescent molecule cybergreen, conducted the real-time PCR, which gives you CT value, and this CT value is finally converted into your log fold change, what you are observing into the published paper. Three important points to be discussed is the primer, how you can design the primer efficiency and primer specificity. Reference gene, we will check that how we can find a good reference gene, and then conduct the relative expression using delta-delta CT value and the Pockels method. First step is to design the primer. Since cybergreen dye binds to any double-stranded DNA product, and its success greatly depends on the quality of the primer which we are designing. Two important points which needs to be considered is the primer efficiency and then primer specificity. So efficiency means that Efficiency means that every cycle, for each cycle, there should be twofold of increase. And specificity means it should not form the primer dimer, or it should not go and amplify the co-amplicon or non-specific binding to amplify the second target. And that can be found using the melt curve. So when we are designing primer, we have done it yesterday using the primer 3 and the oligo analyzer software. We need to consider that the amplicon length should be around 120 bases. It can ranges from 80 to 200 bases. Why? If you are increasing the size of the amplicon, the fluorescence intensity gets saturated, and thus it is difficult to actually calculate the real-time amplification. The primer length should be 18 to 20. If it is too small, then that will cause non-specific binding. If it is too large, then it will have higher melting temperature, and then subsequently problem in the binding to the or annealing to the target gene. The temperature should be around 60 degrees Celsius, that's the melting temperature, and both the forward and reverse primer should have a melting temperature within 2 to 3 degrees Celsius. The GC content should be around 50. It means that if the GC content is too high, again the melting temperature will increase, and if the GC content is too low, then that will lead to the non-specific binding because the melting temperature is too low in that case. Also, since the primer and the amplification starts at the 3' end, the stability at the 3' end is very important. Higher the value at the 3' end causes the better efficiency of the primer. And also we need to keep in mind that the primer should not form the dimer, whether with the self. It means that primer 1 itself is making self-annealing. This is the example which you can understand the self-annealing. It should not form loop. It means that some bases within the primer sequence should not form a loop, or the first few bases should not be the complementary to the last few bases. And the third important point is that it should not cause a heterodimer. It means the forward and the reverse primer should not be complementary at more instances or at more bases, so that it forms the dimer. Also yesterday I emphasized that we need to take care of the delta G value of all the reactions. If the delta G value is more toward the negative side, it means the chances of these reactions to happen is much higher. So we need to remove bases from either of the two ends so that the del G value decreases, increases to the 0, right? You already have a copy of sequences in the Google Drive, which you can try by yourself using primer 3+. Activate the qPCR setting. You can cross check, and in the tool or in the primer 3, you have all the explanations given for each and every point. Once you select the first set of primer, you evaluate using the oligo analyzer, and then in oligo analyzer you will be able to see the self-annealing, loop, and heterodimer by primer 1, primer 2. So these are the points which you need to take care when you are designing the primer. Now, once you have designed the primer and you have conducted the PCR, real-time PCR, we need to be critical that every tenfold dilution, it should be 3.3 cycle apart. And if that is happening, you can plot a linear regression curve, which we will be doing it just maybe in a couple of minutes, and then calculate the slope value, right? Based on the slope value, your efficiency can be calculated. And then the second point is the primary specificity. It means that what we need to understand is that the melting curve or the melting point of any amplicon is a very characteristic to one particular amplicon. Let us suppose 100 base amplicon is there. So it will definitely have a melting temperature which varies from the amplicon with a 50 base pair, right? So in case if you have targeted non-specific bindings in your PCR, you may get something like this with 2 peaks. It could also mean that there is a variation in the genotype, but that we are not considering right now because we are focused on gene expression and we are thinking or we are assuming that the primer which is prepared on the gene specific specific for the gene which does not have genotypic variations. So in this case, if you are getting a single peak like this, it's a normal good denaturing curve, melting curve. If you are getting a melting curve at around 62 63 degrees Celsius, it means that, and then if you see the intensity in other curve, you will see that 1.4, 1.8, 1.4. If you are seeing zero point 0.1, it indicates that it is a primer dimer and it is not the specific amplification. However, in this particular case, you can see that there is a small dimer, and in the fourth example, either it could mean that there is a non-specific binding, uh, otherwise it's a dimer, protein, primer dimer along with your gene of interest. So these two things which we need to take care. Designed the primer and you have conducted the PCR, real-time PCR, we need to be critical that every tenfold dilution, it should be 3.3 cycle apart. And if that is happening, you can plot a linear regression curve, which we will be doing it just within a couple of minutes, and then calculate the slope value, right? Based on the slope value, your efficiency can be calculated. And then the second point is the primary specificity. It means that what we need to understand is that the melting curve or the melting point of any amplicon is a very characteristic to one particular amplicon. Let us suppose 100 base amplicon is there. So it will definitely have a melting temperature which varies from the amplicon with a 50 base pair, right? So in case if you have targeted non-specific bindings in your PCR, you may get something like this with two peaks. It could also mean that there is a variation in the genotype, but that we are not considering right now because we are focused on gene expression and we are thinking or we are assuming that the primer which is prepared on the gene specific specific for the gene which does not have genotypic variations. So in this case, if you are getting a single peak like this, it's a normal good denaturing curve, melting curve. If you are getting a melting curve peak at around 62 63 degrees Celsius, it means that, and then if you see the intensity in other curve, you will see that 1.4, 1.8, 1.4. If you are seeing 0.1, it indicates that it is a primer dimer and it is not the specific amplification. However, in this particular case, you can see that there is a small dimer, and in the fourth example, either it could mean that there is a non-specific binding, and then or otherwise it's a huge dimer, primer dimer along with your gene of interest. So these two things which we need to take care. Okay, so now how we calculate the primer efficiency? Yesterday we did some steps. Let's do both the things parallelly. So I hope you remember yesterday that we exported the result from our Quant Studio software and copied the target gene, CT value, as well as the quantity of the cDNA which we have used in our analysis or in our sample. So this is the result. So you have the gene name, you have the DNA concentration and you have the CT value. Now, let us go and check that how we can make this curve. So same gene, same gene, you have the cDNA concentration. Now the first step is that cDNA template, you conducted a PCR with a cDNA template of 10 to the power -1, -2, -3, -4 dilution, or whatever you have taken in a fixed serial dilution series, right? Export the result which you have done. After the export, you can copy this data over here, which looks something like this, which I have also highlighted in the PPT. The third step is that convert the cDNA into the percentage. And how you can do that? You can assume that the highest concentration, which is 1 in this particular case, can be chosen as 100% of the template, right, or the cDNA concentration. Similarly, what will happen if 1 is 100%, then 0.1 is 10%. So you have sample and triplicate. So what you did is that in the step 3, if you remember, if 1 is considered as 100, then 0.1 is considered as 10, because 100 or 0.1 is 10% of 1, right? Similarly, 0.02 is 10% of in this case, I have not taken 10 to the power -1, -2, -3, -4, because many times practically if you are going with -4 dilution, then the amplification is not done because of the very less template. So what I did here is that I have taken 0.02 microliter or 0.02, that is 2% of the template, and finally 1% of the template, which is also going into the increasing order, right? Now the fourth step is to convert the cDNA percentage into log fold, because if you remember well, what happens is that you need to find a linear, slope of the linear regression, and for that you need to convert your cDNA concentration into log, so that it gives you a linear curve. Otherwise, the mathematics or for the curve, non-linear curve would be more difficult to understand. So what you do is that you convert the log of percentage. So in the Excel, there is a function of log, which you can just select the column of the cDNA concentration and then click on it, right? Similarly, for log 100 is 2, log 10 it's 1, and so on, right? After that, in the step 5, what you need to do is to plot this log value with this CT value, and then you will get a graph something similar to this, right? In this, you click on any data. As for example, I am clicking over here, and you can add a trade line. Once you are adding a trade line, there are two options in the new dialog box. One is to display the equation and display the R squared value, right? So this is what you are getting. This is what you are getting, right? Now, this particular slope value is -3.540, which you write down in the F column, F4 column, right? Using this efficiency, using this slope value, we can calculate efficiency. If you remember, it's a log value, right? And it's a log graph, right? Now, in order to convert the log graph, the value of the log y-axis, there is a function having a formula that 10 to the power, and since it's a linear regression, 10 to the power -1 by slope value. So that is your sixth step, -1 by slope value. If you see here also, you can see 10 is to power -1 by this F4, that is the slope value, right? And then you will get the efficiency. If you remember what we discussed in the 2 slides before, if it is 100% efficiency, there is a 2-fold increase. However, in this example, what we are getting, this primer is having less efficiency than 100% because in every each cycle it is able to amplify, or it is able to it is not able to double the template or the gene of interest. It is making 1.91 times. So how we can convert the efficiency in percentage, that is the last step, is that you use a formula that whatever efficiency has came here, you subtract it with -1 and then divide by 100. That will give you percentage. Now this particular example says that this primer is 91.61% efficient but not 100% efficient. We have to keep in mind that anything between 90 to 105 or 110% efficiency, we can use it in our analysis. Another thing to keep in mind is that for 91, definitely you will use 1.91 in the relative expression formula, which we are going to see next. Similarly, anything above 100% should be used as 2, but not more than 2, because any machine or any enzyme cannot have efficiency of more than 100%, right? So that we need to take care. If it is varying too much, like less than 90%, 85, 86, or let's say 80 or 65, then we should not use. Similarly, if it is going beyond 105 or 110% efficiency, then we should not use that primer, and instead we should design a new particle. So I hope that this is clear now.